This is the reaction I was talking about while you couldn't see it, and I will just throw my link there. So we're going to take this two-position alcohol and turn it into a ketone through an oxidation with a concomitant reduction of our cofactor, the MDH, showing the nicotinamide ring here, and having that hydride transferred onto it as part of that reduction process. And so how do we package this into a three-dimensional structure that allows for this to happen efficiently and potentially with regulation attached to it? So things about malate dehydrogenase. So we can go and find a ton of malate dehydrogenase sequences in things like UniProt and other databases. And what we find is that this enzyme, because it is doing an essential reaction for life, it is found across the phyla, and that on average, these are around 300 to 350 amino acids long, and they are about or a little over 100 structures. Unique structures, there's around 50 that have done experimentally, but of course with computed structure prediction such as AlphaFold or Rosetta Fold, now you have access to basically input a sequence of your favorite malate dehydrogenase from any organism and get a fairly decent model to start with, which you can then validate through experimentation. So today I'm going to focus on just those structures that have been done experimentally, although you do have access to AlphaFold, et cetera, to do anything that is of interest to you that is not in things like the protein data bank. And so if we look at the sequences of malate dehydrogenase across the known structures, we did in this review with Chris, we had done a large cluster alignment of all the unique sequences, and I'm just showing a few that are representative. So at the top, we've got a thermophile that's a bacterium, E. coli MDH. This one is an archaea, a yeast, a human, and then one from the plant world. This is watermelons, and from the glycosomal organelle. And so just showing those alignments of those larger ones as a subset, what we find is that we have some conserved places, and we also have a lot of variation. In fact, it's fairly common to have only around 20% sequence identity, sequence similarity across MDHs, sometimes even in the same organism. So cytosolic and mitochondrial MDH in humans is quite different if you look at it at a sequence level, but overall the structures are near identical. If we look at this representation here, we have the secondary structure showing above the sequence, so that you can see we have in the first half of this molecule, this beta alpha continuation. We're going to talk about the fold and what that turns into. In that first half, we have conservation of sequences that are dealing with the binding of ligand. So at the beginning here, we have some glycines that are conserved at this position and this position that dictate the cofactor binding site, and it's a motif that's found across nucleotide binding components. And I'll show you that in the structure in a couple slides. Then moving on to the second line here, we have another area of conservation, and this is again associated with the active site. And we're going to have come into play three particular arginine residues that are be important for coordinating the substrate. And so the first two appear here in the sequence. And then we have this asparagine, which I think is underappreciated, but is vitally important in a number of ways. And then moving down, another asparagine, which I'll talk about in the structure. And finally, the third arginine that is involved in substrate coordination. Coming down to the third line, now we're moving into a slightly different setup where we start to go beta, beta, alpha, alpha in terms of secondary structure. This is indicating a second domain, which is going to be involved with substrate binding. And in this area, we have the conservation of the histidine that'll be involved in the catalytic mechanism. And so if you do these sequence alignments, what you can identify are areas of conservation, but also you can identify areas that are dissimilar, which might be even more interesting and allow you to identify cryptic sites that you can capitalize on in the studies that you want to pursue. So based upon that primary sequence, now we have the tertiary structure of MDH. I apologize, I worked on a movie last night, but my output was slower than my eyes could stay open. So you're going to get still pictures today. What I am showing you here is the tertiary structure starting at the N terminus here, and it's going to do a series of beta strand, turn, alpha helix, beta strand, turn, alpha helix. This is forming the first domain, which is the Rossmann fold, coming down until we get to kind of this pea green color here, where then we transition into the second domain, which then goes through the yellows into the oranges, and then finally ending in that long C-terminal helix, as is shown in the structure, but which Chris alluded to is more probably dynamic than we give it credit for. Along here, I'm showing you the cofactor binding that occurs in one of the domains. And then at the base of this helix is where your substrate is going to bind. So I'm going to walk through some of the structure of this tertiary component. In that, we can separate it into an N-terminal domain known as the lactate dehydrogenase N-terminal domain because it's shared across the related families of lactate and malate dehydrogenases. It is forming that alpha beta alternating sequence, also known as the Rossmann fold, and that along the edges of the beta strands here on the loops is where the nucleotide is going to bind. And we'll show that in a couple slides showing in detail how that's going to occur. And then, of course, the substrate binding domain is shown down here in the blue, and we'll walk through the details of that. So in the first domain, the N-terminal domain that is the lactate dehydrogenase N-terminal domain, I'm labeled now the secondary structural elements where this is the N-terminus at this position. I'm also highlighting here that the nucleotide that's binding along the edge of the beta strands and alpha helix loop connectors in this kind of minty green color, all of the residues that are important for that binding of the nucleotide and the selectivity. So at the top here, we have the adenine ring, then the ribose, the two phosphates, the second ribose, and then the nicotinamide here, poking down and leading into the second domain, which is going to bring the substrate to the party. Also within this region, in addition to binding the nucleotide domain between beta 4, leading into this long extended loop and heading into then alpha 4, I've colored it in red. This is the area that is going to cap over your active site. So this is known as the active site loop. Some people call it the dynamic loop. Some people call it the mobile loop. But it is a place where there's an opening that leads into the active site, which then can take on several conformations, as Chris alluded to in his short intro. So we're going to talk more about that in a moment. So this is kind of generic, and I wanted to show you a little bit more detail. So I've done what's called a link plot on that shows in 2 dimensions the binding interactions between a ligand and its protein. And so this is just the adenine ring at the top, ribose, the phosphates, and then the second ribose and the nicotinamide ring. You'll note on here that you have these little things that I call eyelashes. They're indicating that it's forming an interaction through an intermolecular force with the surrounding protein.
Jess, I can't see the, I just see the, I don't see the eyebrows. I don't know.
Oh, they're kind of tiny on this one. There's going to be some bigger ones that pop
Okay.
up.
I'm
Yeah.
used to seeing them and they're much bigger. So, okay. I was just wondering if it was
These are
not
little
showing
tiny
everything.
ones, just
Got
around the
it.
little atoms.
Got it.
And it's just showing that it's reciprocating the interactions that I'm going to show you later.
Okay. Yeah. Just wanted to make sure if there was something that wasn't showing.
Thank you for checking. All right. So first thing we have are direct hydrogen bonding interactions. And so we have this glycine here is in that motif that's used to bind nucleotides. And so you can see that it's forming with its backbone amide a hydrogen bond to one of the phosphates, for instance. And its neighboring isoleucine is also using its backbone to do that. Then I find that there's this fun area. The one I'm showing you is from E. coli, so the numbering is from that. This glycine 78 is at the beginning of the active site loop that covers over the active site. And so at the beginning, there's the backbone is able to sense, is there a nucleotide there? And then at the far end of the active site loop, there's an asparagine that kind of caps off that active site loop area. This asparagine is also using its R group amide to form a hydrogen bond now to the ribose ring. And so in that way, the active site loop has a direct trigger of interacting with and sensing, is cofactor there? Can I make an interaction? This asparagine 119 is that other asparagine that's highly conserved. And you'll see here that it's making interactions also with the cofactor. And I'm going to show you that it plays another role that is equally important in terms of substrate. And so the readouts from these guys, when we're looking at interactions, you can get an idea of how is the three-dimensional structure of the protein sensing whether or not cofactor is present. None of the residues I've highlighted here are involved in catalysis. They're all involved in orienting and binding to the cofactor. And so this is just showing you how those residues are sort of giving the cofactor that bear hug. So here's our cofactor here. And the residues that I've highlighted so far are in kind of this purpley blue color and showing how they kind of surround and hold that guy in the right orientation. And then the final one I want to point out here. So these are the big winkies so that you can see these are the intermolecular forces that the rest of the protein make with that nucleotide. And what I want to point out here is this aspartate 34. It is in close proximity to the ribose group that's attached to the adenine. And what it does is it's looking for and interacting with the hydroxyl groups at the 2 and 3 positions. And you'll see in this three-dimensional rendering that they face in and actually have the potential to do hydrogen bonding there. And it is sensing that whether or not those are hydroxyl groups, or if the 2 position is phosphorylated, such that this would use a cofactor NADP. And so I'm going to show you now the difference between those. Across these winkies that you see here that are showing those intermolecular forces, you have quite a few interactions that are also emanating from the dynamic active site loop, and they're forming contacts here. And so that closing of the loop and having it come into close proximity with the active site is helping to stabilize the interaction between the ligand and the protein. So this is just a repeat of that one I showed you before, kind of pointing out that aspartate that's close to the proximal proximity of this ribose hydroxyl groups. And in plants, they're going to use the NADP, NADPH cofactor. And what we see is that then that aspartic acid is replaced with the glycine, which will allow for much more room of that phosphate group to come in. And in addition, you're having additional amino acids come in and form direct interactions and stabilizing that negative group that comes in. So we have some serine and then some backbone ones from other residues coming in. The difference between these two in terms of the colors of the winkies, the blue ones are shared similarity between the two, and the reds are unique for NADPH binding on the right-hand side for the plant and NADPH versus the NADH binding on the left to most of the other structures of MDH that are known. So that's sort of an overview of the NADH binding domain. So the gray guy at the top. I'm going to shift over to now talking about the substrate binding domain and some interesting structural features of that. So I apologize, I changed the orientation, but there's this cool feature in MDH that is just one of my favorites, and I can't pass up the opportunity to talk about it. So I kind of nerd out when I get to talk about secondary structure to my students. Oh my God, I love it. Like hydrogen bonding, intermolecular forces, look at circular dichroism, Ramachandran plots. Yeah, they're all my shtick. But in here, look at this guy. So you have this alpha helix here, and then you have this opening, and then it continues. Holy smokes, how many times have you seen an alpha helix who does a 90-degree turn? Isn't that cool? Oh my gosh, MDH does this beautifully. So you have this beautiful alpha helix coming along, and then you break the hydrogen bonding pattern of the alpha helix, and within there are some crystallographic waters we can observe that maintain hydrogen bonding such that this guy takes basically a 90-degree turn. And nestled right in there are the interactions that form the substrate binding pocket. We do have some MDHs, there are a couple of them that actually don't have this bend, and they're using a little bit alternate fill in the active site to accomplish binding to the substrate. But most of them have this very unique bend in this alpha helix that is going within the active site. All right, so again, I've kind of colored in that, in this case, a cyan color, every residue that's sort of touching the substrate and how it's holding it and oriented in the active site. And then I'm going to show you again a leg plot where I've got oxaloacetate shown here and its ability in this two-dimensional rendering to interact with different groups. And so I'm going to start at carbon 1 here. This carboxyl group is going to be coordinated to, and in this case, I'm showing you the human malate dehydrogenase. And so the numbering, I apologize, has changed. So this arginine here that is conserved at the back of the active site is coordinating to the carboxylic acid group here and kind of pulling that OAA into the back of the pocket. You have your catalytic residues here. So this is your histidine that is going to abstract or donate a proton, and it is being modulated by this aspartic acid here. At carbon 2, this is where you have your an OAA, the ketone, which will, in this case, accept a proton to become reduced. It is being coordinated by an arginine that is on the active site loop that can move, so it can pull in and coordinate to that. Look, an asparagine. This is that same asparagine that was interacting with the cofactor binding. So now you have this guy doing double duty. So this guy should probably still be coordinating to the ribose hydroxyl of the cofactor, but when OAA is present or malate, the R group amide can interact with potentially the either alcohol group or carbonyl oxygen of the ketone if you have OAA there. And so this guy is a main player in our work. Carbon 3 here, kind of boring, does intermolecular forces to other things around it. And then finally, the fourth group with the carboxylate, it is interacting with another arginine that is on the active site loop. Apologies. And then you have some other surrounding amino acids that are able to interact, as well as the cofactor is going to make some interactions as they're stacked in the active site with one another. And so this should give you some ideas of, I'm showing you more or less what we call the first sphere, or those things that make the direct contact to the active site players. But you also want to think about other amino acids that are making additional contacts with those. And we'll talk about how that may be occurring and transmitting information. And so Ellis, I think, might tomorrow talk about one project that is looking at the second sphere of amino acids, which are helping to make sure these guys stay in the right orientations. So that's your tour kind of the tertiary structure of the two domains that are present and then their ability to bind. Now, if we look at the quaternary structure, I have to tell you, malate dehydrogenase does not work as a monomer. It is very inefficient in that oligomeric, non-oligomeric state. And so in the wild, what we typically find are dimers, which the human, both cytosolic and mitochondrial, tend to form. And then we have also tetramers that are available to us. These tend to be the archaeals and the apicomplexans tend to form more tetramers, although there are some other examples. Also note that a lot of lactate dehydrogenases, a highly related dehydrogenase, can also form tetramers. And so there's some crossover there in terms of evolution of retaining that tetrameric structure. So if we have, in the dimer, of course, we have this single interface here. You can, if you look right here, this is the center of the twofold, and you'll see that this alpha helix is this alpha helix, but rotated at 180 degrees. So you can see that twofold symmetry. In the tetramer, what we do is we take that dimer, and it's a little bit turned, swiveled to the right, and then we stack another dimer on it, creating additional interfaces. And so you have a BCAD interface that you can think about, interactions, as well as there are few, but there are some interactions across the AC and the BD interfaces that can be thought of as stabilizing some of these interactions. All right, you guessed it. I was going to show you another leg plot of what these interactions look like. And so in the dimer interface, we can look at across this dotted line who is interacting with who. And so if we look at it from one perspective, in terms of the B is on the top and the A is on the bottom, we have some hydrogen bonding potential. So we have an aspartic acid that is able to interact with the backbone of several amino acids, as well as an R group of a lysine to do a charge-charge. And then on the B side, we have this histidine which this is kind of cool because this is pH sensitive. So if you think of protonated versus non-protonated, an interaction between this histidine to this glutamate is much stronger when they're carrying charges, but is still present even in a polar, but could weaken the interface. Here's a key player here at the interface. This is the arginine. This is E. coli numbering for this particular example. This is the arginine that is at the back of the active site that is coordinating and pulling your substrate into the back of the active site. It is also capable of talking to the interface of the adjacent subunit. So again, you've got some sensing here across an interface of, is your active site full? Does it have a substrate in it? I could potentially communicate that to the other neighboring subunit, setting up a relay system between the active sites. What's interesting about the setup here, if we're looking from B to A here, you can do the same and look at the second configuration, and it is more or less mirrored between them. So there are a lot of interesting things you can look at subunit interfaces. Some of them change somewhat. This is the E. coli one, but you can look at different organisms. Some of them are fun to think about in terms of thermophiles or halophiles, etc., that are maintaining these subunit interfaces, but under conditions that might challenge them in terms of heat or salt concentration and compete for some of those interactions. All right. So that's our active site and our players and what is going on in this region. I wanted to spend just the last few moments talking about this active site loop and its dynamics. So Chris alluded to the ability for it to have some movement and take on different conformations. And so as part of our look at structure, this one was done by Ellis, actually did these comparisons across different structures. And what he noted in aligning the dimeric structures is that on one side, we have this active site loop, which is kind of in an up or open position. It's not exactly the same, but they're all in that open position. And then over here, this second active site loop in the adjacent dimer, you can see they take on kind of two different conformations. There's one that's very tightly closed. The blue one's very close to it, coming in contact with this green cofactor / substrate pairing in the active site. And then there's one that is not quite fully open, but it's not fully closed. And so Chris alluded to there having multiple conformations here. And so what can we look at in terms of the structures and kind of define what we're seeing and what's available to us experimentally? So this is part of the work that Chris did for the review that was published, where he took all of the known experimentally determined structures of MDH and did a principal component analysis of comparing atomic distances, etc., between this database of structures. And what came out of that was basically three different clusters of structures. And so we've got a cluster up here, a cluster here, and then a cluster here. And that we could look at them properties that we could define and see how they distributed across those clusters. And what was interesting is that when we looked at oligomeric state, in the blue, we're showing the dimers, and the dimers all cluster either in this guy or in this guy. And the picture that Chris showed earlier of the active site loop and slightly different conformations, that was basically the separation between these two different states. What I found interesting as well is that these guys are primarily tetrameric, except for a few examples. And these three examples are either archaea or apicomplexans, which is where we also find most of the tetramers. And so there may be some holdover or differences in some structural elements that we haven't yet defined that allow us to understand why some dimers are separating out down here. We can apply other criteria to these PCA clustering. If we look at phylogeny, do they separate based on how evolutionary distant they are? It doesn't appear that, although all of the archaea tend to be in this cluster down here. And then also with differences in the dynamic loop, are they being mediated by having substrate present, nucleotide present, or having both present? And it looks like not really. They can be in the apo state, which is the purple, or in the black, which says there's something bound to it. and they divide between the two. And so suggesting again, it could be just how we've captured it and may not be totally reliant on just having the ligand present there. So it's not an easy justification for why we have these different clusters, especially across the dimeric states. So there's some great questions in there and more things to delve into in terms of structure. So one thing that Chris identified and he alluded to earlier was how can we talk about kind of how these changes may be mediated between them. And so I know this one's kind of busy, apologies. But what I really want to get at in this structure of malate dehydrogenase, so this is a dimeric structure, and we have one monomer here, and the active site is filled. And then we have a second down here, and the active site is filled with ligands. And I want to talk about what's happening across the interface between the two subunits and how we can communicate things. So I'm just going to blow up the area of the two active sites in the interface. And so that's what's shown on this side. So if we look at this guy here with cofactors bound, and that's in the, in the blue, you'll see that the active site is completely closed and clamped down. And in doing so, then it can interact with this alpha helix here that is close to the interface. And this alpha helix here can then interact with an alpha helix across the interface, alpha 2. Alpha 2 coming out of it has that amino acid, which can coordinate to the ribose hydroxyl groups at the 2 and 3' positions. And so then you have kind of a direct connection, RU closed, pushing on the interface, across the interface, and into where the cofactor potentially could bind in the second active site. And is that communicating this active site is full, the other should be empty, or vice versa, allowing for the enzyme to load and unload ligands properly. And then in the orange is showing when you're not fully closed, this active site loop does not push on this guy, which then allows for a reorientation of the alpha helix across the interface, and then a reorienting of something that could sense whether or not cofactor was present in the adjacent active site. And so these are some of the interesting kind of structural analyses you can do with different MDH structures, allowing for you to come up with ideas for your cure. All right, with that, many thanks to the Bell Group, especially my partner in crime, Alice Bell. And these are some of my fun students making their silly face at our graduation party. The MDH Cures community, and of course,